Thank you, Christian, for the kind introduction. Hello, everyone. Thanks for joining us today. As Christian said, my name is Franka, and I'm going to talk about chemical space exploration today. And I want to start out by what is chemical space. And there is this relatively old publication where people said chemical space is a vast amount of molecules, which goes up to 10 to the power of 63 molecules roughly, and they say these are drug-like molecules, so really the stuff that is interesting for us and not just fancy molecules that might not be working in the human body or on any other target. And when we look on how far we have come so far, it's relatively surprising that we're still somewhere around 10 to the power of 6 with what's purchasable on e-molecules. And there are approaches to collect more molecules in virtual databases like the real database or the zinc database, for example. And there is always a cutoff at roughly 1 billion molecules. And the reason for this is that all those classical databases are enumerated databases. And enumerated means, for those who are not aware of this word, that there is a chemical and an order number written down one after the other in your computer. So a really long list of molecules and order numbers. So really long list means, of course, lots of data. And when you want to search in them, it takes quite a long time. And with current computer powers, there's just no good way of handling more than 1 billion of compounds with these classical approaches. And we have developed a very different approach to tackle more compounds by doing synthesis in the computer. So in silico synthesis, teaching the computer chemistry and giving it then a list of building blocks, telling it how to combine it. And this makes it much more fast to search, and we can access much bigger spaces. So coming to how it really works, we teach the computer chemistry is what I said. So for example, we look here at the Suzuki reaction, and we look at the minimum which is necessary to really make it a Suzuki reaction. So we have a carbon attached to a boronic acid, and we have a carbon attached to a leaving group. And on the product side, we would have a carbon connected to a carbon. And if we feed now a database into computer with reagent, it will automatically find the reagents which match, and it will clip off those leaving groups which not survive on the product side and attach little linker to them, which are depicted here by those Lego bricks. And our product side is never really there. It's basically still our building blocks, which are together with those Lego bricks, because you know all how this happens. I, of course, present much more building blocks to the computer than only those two. So the moment where I add more reagents, I get all the cross products, and my product side would completely explode. So we basically ignore this right side, but just keep the left side, our building blocks, and how it's compatible to each other. So imagine you have 1,000 boronic acids and 1,000 of those carbons with a leaving group. That would make already 1 million products on the right side, but we don't do this. We keep the 1,000 and 1,000, so 2,000, and therefore have access to 1 million by just looking at this side. And this is an extraordinary efficient approach when I teach now the computer the next reaction. On the one hand side, those reagents can reoccur. So our boronic acids are able to react, for example, in this multi-component reactions. So I have stored those thousands already. So I can reuse those building blocks with the Lego bricks. That's the one advantage of making this so efficient. But the other thing is, if you have something like a 3-component reaction here, like this petasis reaction, you just add another 1,000 and another 1,000, and then you don't have 1 million, but you will have 1 billion products already with 1,000 and 1,000 and 1,000. So keeping in mind that we have now here 3,000 plus the 1,000 from before, so with 5,000 building blocks, we can access already billions of molecules. So summing this up is basically this building of a space in terms of collecting your reactions that are out of interest for you, and then you take the building blocks and pre-process them to those little Lego building blocks, as I showed you before. And on the right-hand side, you will start a matrix from what you've learned earlier from those reactions. So I said yellow fits on green. There you have this in a matrix, and the green was fitting on the red as well, and the red was fitting on the blue as well. So you have this one big matrix, And on the other hand side, you have your building blocks with those little linkers. And of course, there can be many linkers. That's why I have all those different Lego bricks here. And with this approach, which we call a fragment space, we can go and create really big spaces. And you see here different spaces that are out there at the moment and are present in the current publications. And when you look at the really big ones here, which are proprietary from pharma companies, oops, you will, there's an animation missing here, unfortunately. So all the ones here have those little biosolvit logo, means like they're created with the biosolvit technology. That's how we can go up to spaces that are 10 to the power of 20, which is the Merck Massive, which is currently the biggest space out there. And those spaces, when you look at them, are surprisingly old sometimes. So we started doing this a long while ago. And the whole technology only got really public interest, when we started creating the enamine real space. Because the real space is a space that's purchasable. And purchasable space is really big. The real space is much bigger than those other spaces. When you compare here the molecules, as I mentioned before, and then you look at all those virtual databases, and now I have to zoom out. I lie here a little bit to you because this is only the last version of the real space. The current version is actually much bigger. It's 13 billion at the moment. So a much bigger source that you can access and purchase from than what you normally are used to. But the question is, how do you search in such big spaces? And because I've said we have those reagents here with the linkers and we have the metrics how to combine them, but we're of course searching for real molecules. And a real molecule doesn't have any Lego bricks or linkers in the middle. So the question is, how do you search in there? Because you want real molecules back and not just Lego building blocks. So we have a style of navigating through those building blocks or finding molecules by using an algorithm which is called feature trees. And feature trees takes your query molecule and translates it into such a tree of pharmacophore nodes. Each of those nodes has a profile behind it. So for example, the piperazine ring here has a particular volume, has a ring count, and has a little pharmacophoric profile. It's hydrophobic, it has a little bit of a donor and acceptor feature, no aromatic feature, and so on. And in this way, we represent each part of the molecule as a pharmacophore node. So it's a little bit fuzzy. And this tree we do take as a blanket, not as a blanket, as a blueprint for searching in the space and coming back with a second tree. You see those bits map onto each other, and there is sometimes no maps, but sometimes there are maps. They're differently connected. And this then retranslates into your hit molecule. And when you compare now query and hit, we've seen that there's quite a few chemists here today. So on the first glimpse, you might not directly see how those molecules are similar to each other, but when you take a closer look, you will see the bicycle is here and then the hit. The amide is still there, it was in the ring before. You have the aromatic core in the middle with a hydrophobic substitution pattern. The sulfonamide is still here. You have here an open chain, alkyl chain. Here you had it in the ring as a piperazine before. So yeah, on the second glimpse, they are similar. And I particularly chose this example because both of them are active molecules on the same target. So this algorithm is able to not only find really close analogs, but it's able to see scaffold hops over different compound classes and still find your active molecules back. Question is now, of course, how do we search in those fragments? And I lied a little bit to you earlier. So there's not molecules with linkers. There's basically mini feature trees with those linkers stored in our fragment space. And when we have a query molecule here, we this is a dynamic programming algorithm, which basically reassembles your query molecule with those individual bits from the fragment space and tries to find the most similar molecule to your query molecule. It's a little bit similar like when you do a sequence alignment where you find parts that match to each other and can have gaps and all the things, as you know it from there. So nice about it is that you can see in the end how those little trees were mapping onto each other. So you have the green bit here. As you saw before, we went through the molecule and analyzed the different features already. And yeah, you see the global similarity between those molecules, but you can see the local similarities as well, which is really nice when you want to emphasize a particular feature, which is possible as well with this algorithm. You can select those nodes kind of beforehand in your query and give them a particular rating on how important they are to you, so that they get exchanged or that they are maintained whilst you're searching. You can access this in two different ways, either as KNIME nodes, where you draw a query or read a file and search it in the space. And on the other hand side, there is a really interactive interface, which is called Infinicy, where you paste your query here and you have a few search parameters if you want to, and you have a play button here which generates you the results, and when you click on those results, you get those mapping images as I described them to you before. And the question is, can I really mine from such a big pool? Because I have to go back to the linear scale on a regular base. Again, this is the zinc database where people get really, really excited when they can dock them. But when you look at the real space, it's much bigger than the zinc database. And the question is, can I really search in there and find something that's close to what I'm looking for? And I'm going to present you here a study around BRD4 inhibitors. It's a pretty easy, straightforward KNIME workflow. There was a publication where they've looked for molecules which inhibit BRD4, and they had found a good set of real binders and good inhibitors. And then they had a set of 14 known actives, and I have put actives here extra in high comma, because those molecules showed a little bit of inhibition on the target, but basically no one could measure an IC50 because it must have been so high that you were basically out of what your assay could measure. So we've taken those 14 kind of active molecules and searched brute force 5,000 similar molecules in the space, docked them with 5 poses per hit, and selected out of the best molecules 32 compounds. And we applied this here on the real space, so we could order the molecules from enamine, and we validated them in the first place in a thermal shift assay. And what you see here is the compounds and their thermal shift at different concentrations. So you see those dependent thermal shifts and hopefully binding on those on the target. But yeah, what I personally love about that slide, and I need to take a second here to make you count, that's actually 32 molecules. Yeah, so all of those molecules that we've selected from those billions of molecules were really synthesizable, which is a perfect example for how creation of those spaces and taking care of the reactions and the building blocks makes a big difference here in this game. And yeah, coming back to the science, the thermal shift assay should of course not be the only thing to look at. So we measured really in an activity assay the activity on the target, and I'm showing you here a few of the best compounds. Their IC50 is here at the back column, so the best compound here has 10 micromolar. And I've shown here the query hit pairs for you. And when you look at those molecules are surprisingly similar. So I still have here the right side maintained the blue bit. I have the amide. I have suddenly a 1 position longer linker between the aromatic core and the amide. And I have a different hydrophobic pattern here. The oxygen is in the furan ring, not a alcohol anymore. So yeah, they're kind of similar when I look at them. That's what my feature tree similarity says as well. When you, on the other hand side, calculate a Morgan fingerprint and calculate the Tanimoto similarity, you will see that the similarity between those two is surprisingly low already. So using a standard similarity search, you might actually miss those pairs, whilst feature trees ranks them all relatively high to each other so that you find them on the top of the list of your similar molecules. I want to particularly highlight here those query and hit pairs, because they're really surprisingly similar. Instead of the methyl group, I have a third butyl / a 3-fluorine methyl group here, and the linker is a little bit different, and the nitrogen in the ring is shifted by 1 position. And when you look at those docking modes, you will actually see that you flip them completely around in the docking mode, because the distance between this nitrogen in the ring and your heterocycle is suddenly in the right position to attach to 2 two backbone or to two amino acids in the protein, therefore bringing up a completely new hydrogen bond and therefore most likely being active, whilst the first molecule was able to only perform or build one hydrogen bond here and therefore not being really active on the target. So I know that those molecules with 10 micromolar are not the particular greatest wisdom of BRD4 inhibitors. There are much more active compounds around. But the interesting bit about the study is how strongly we could streamline the process. I showed you this 9 workflow. This was really not a big deal, putting 40 molecules, straightforward searching, docking. So the whole design cycle kind of part was just below a week. And the ordering from enamine and the synthesize took less than 3 weeks, and the testing another 2 weeks kind of with the 2 assays. So we're here in below 6 weeks, going from a few inactive or kind of active molecules to molecules there you can really measure your IC50s from and enrich your SAR by a couple of molecules to get the first QSAR models out there. This is another application example from the people from Merck. They apply their massive space in a numerous drug discovery project. This is a nearly 2-year-old slide now. By then it was all relatively new, but they had already applied it to 12 different drug discovery projects where they did 1 to 4 searches in their space in each of those drug discovery projects. And for each of the search, they've selected 4 to 60 compounds, which they synthesized. And again, they could state as well that the feasibility of the molecules, so that they could really synthesize more than 80% of those molecules. So they sum it up in speeding up their drug discovery process by a factor of 2. And when they ordered the compounds from the real space, they could say that the cost for developing a compound was 10 times cheaper as well compared to their in-house synthesis pipeline. When they applied the massive space, which is built on their in-house chemistry reactions plus their building blocks, so you know that Merck is the owner of Sigma Aldrich, so EMD Serono is the name of the whole company. So they have a massive set of in-house building blocks as well. So what they find in there has a really, really high intellectual property for them. And yeah, so this is kind of the first part of my talk, saying the searching in those spaces works. And Christian has now another question for you.
Yes, indeed. Thanks for this first part, and thanks also for all the questions that you put in the question box already. I try to, answer them as Franka is flying through this presentation. Yeah, after having presented kind of the approach that we have used very successfully so far, I would like to ask you with this poll, what do you miss most when you see us here working with chemical space, or see yourself working with chemical spaces? We have this method that allows similarity searching with this particular pharmacophore-style similarity. But of course, people have asked us, can I query such a space in a different manner? So what is, in your opinion, most important and should be the focus of our attention moving forward? Okay, and I see answers to my poll are coming back. That's great. Thanks for sharing this. And again, coming back to those questions, yeah, keep typing them into the questions box while Franka is presenting. This way, we don't disturb the presentation, and we can address the questions either on the side or in the Q&A session at the end. Okay, I see so far 50% of you have participated in the poll. It would be kind if a few more people give their vote. What do you think is mostly missing? What would you like us to develop next? Substructure search, standard fingerprint searching, some sort of fragment evolution in the context, I guess, mostly of fragment crystallography, docking, and other. Okay, that's great. Now we have 2/3 participated in the poll. Maybe a few more clicks.
Christian, there are a few messages in the chat box saying that the poll doesn't work for everyone.
Okay. Yeah, in the first poll, I also had one. So for those of you, maybe we simply do it this way. For those of you who cannot participate in the poll, and I apologize for technical difficulties, just feel free to also give us that feedback in the chat box or in that question box, and then we can take it into account later. Okay, with that, I'm closing the poll in 5, 4, 3, 2, 1 now, and share the result. So apparently, very head-to-head competition between docking, winner here, fragment evolution, also in the 3D design arena, I would say, but then also of strong interest seems to be the substructure search, and others side by side with standard fingerprints. Again, maybe I reach out to those, last but not least, 10% of you who clicked on other, what you would like us to do in the future with spaces, or what you would like to do with spaces. Okay, and with that, I hand over back to Franka. Sorry for the interruption, and let's go on with the presentation.
Thanks, Christian. Okay, so I showed you that the standard established searching with feature tree in the spaces works, but basically feature trees doesn't have any 3D information. And we were wondering, can we use those vast spaces in 3D structure-based designs? Many of you have clicked docking before and would be interested to dock those spaces. So we are working on establishing a method that we call chemical space docking. And it works in a few steps. In the first step, we place building blocks. So I showed you before that our fragment space consists of those little building blocks with their Lego bricks. And in the first step, we dock them into the binding site. And yeah, we get different solutions here. We have nice hydrogen patterns where the linker is in a good position. We have this fragment here where a linker is pointing directly into the subpocket, so not much room for adding the next part of the molecule on it. And so we have a few steps of manual filtering and visual filtering as well, where we select the building blocks which are in a good position. And in the next step, we take those and look into our compatibility matrix again, because for the green one here, for example, we know the yellow one can be attached and so on. So in the next step, we enumerate the molecules according to those chemistry rules for the full molecules. So taking the green example here, we have it here, and now we attach all the ones with the yellow Lego brick and template-based dock them into the binding site. And there is already one automatic filter, so to say. If you can't place the added fragments somewhere into the binding site, you filter them out automatically, whilst the other building blocks which fit are template-based docked into the binding site. That means the original fragment stays in place, whilst only the new part is wiggled around a little bit and flexibly fitted into the binding site. And so we generate a full list of full molecules, so complete, not only the fragments with the building blocks, from which we can select. And there are numerous applications now where we have done this on different targets, which I'm not allowed to disclose. But we discovered a few challenges and solutions on the way. So the first is, of course, where is your linker in the binding site? I've showed you that when it is somewhere pointing towards a sub-pocket. That's not really helpful. But even if there is space, it might not be space to grow in this direction. Or on the other hand side, if it points straight out to the solvent, there is no room for creating a good interaction network with the fragment you add. So we're still optimizing, filtering out those linkers of the fragments in the first step. Then when you look at those fragment docking poses and only sort them by their score, you would see a table like this where you have different estimated affinities. They're actually really similar, all somewhere in the low micromolar area. But when you look at the molecular weights of those molecules, they vary quite strongly. And therefore, we pay a lot of attention to the ligand efficiency and select fragments which have a good ligand efficiency instead of just an overall good score. And the vast size of those enumerated libraries. So in those enumeration step, you go up to quite a few millions of molecules. And the questions, of course, how do you make this nicely available on the one hand side on the interface, but maybe have a REST API to a powerful computer where you can search and dock those big spaces on your server and come back to your interface for picking the molecules that are out of interest for you. So as I mentioned, there are a few application stories around that I'm not allowed to disclose, but I recently did some voluntary work on those COVID structures from the diamond light source. They had crystallized 17 non-covalent crystal structures of fragment binders in this main protease of the COVID. And these are the fragments that you're looking at here that they have crystallized. You see they're all clustered in the middle of the binding site. They're not really exploring sub-pockets here and there perfectly. And all of them are relatively small and have quite similar structures. And what we did is we applied our chemical space docking approach, and we selected one of those crystal structures as a starting point and docked all the fragments that span up the real space into the crystal structure and selected only the best 100 of those fragments. And I have this rhombus here in the back, which is a little bit a representation of how much data you have in those individual steps. Selecting one crystal structure is, of course, little data. Those fragments are not too many, and you select only the best 100, so still manageable data. But from those 100 fragments, when you apply those chemical rules, come out 1.7 million enumerated molecules. That's surprisingly much when you think it's only 100 building blocks that you're looking at there for the first step. So those 1.7 million molecules were template-based docked on their originating fragment, so 100 different templates, so to say, leading to lots of docking poses, and scoreable out of those were 2.2 million then again. As I mentioned, some of them are not able to be placed. And then we inspected the best 50,000, so we sorted the molecules by score and selected 13 candidates that we particularly liked. We have submitted them to this platform from the Postera people. So if any of you is interested in what type of molecules came out of this, feel free to follow this link and look it up on the Postera website. And if you look at the 13 different molecules that we have selected here, that's basically an overlay of all of them. You will see that they're nicely exploring those sub-pockets on the site. They do share common features here in the center of the binding site because there's lots of action going on here, so which is of course addressed by the molecules that we found, but they grow out in very different directions as well. So you have a really thorough exploration of your binding site in a surprisingly easy to achieve manner. And just one molecule I want to show here as an example is this sulfonamide molecule, which has a good exploration of this hydrophobic pocket on the left. Has a nice interaction pattern with the central backbone here of the protease, but as well does an interaction here with the histidine in this rather polar side chains, plus an interaction with the gatekeeper here on the top. And yeah, I mentioned before that we applied the real space for this study. So when those molecules come out, they come out of the search with such a code down here. And this is basically an order number, which I pasted into, or which I can paste into an email and send to the enamine people for ordering those compounds and getting them delivered, hopefully within the next 3 weeks. So to sum this part of my presentation up, we can say that chemical space docking is able to explore those vast spaces in a 3D fashion, structure-based drug discovery manner, and it leads to purchasable compounds, and it's really, really time efficient and money efficient, as those compounds are surprisingly affordable in the end. Plus, the whole thing doesn't take too long to run. And it doesn't take too long because at the moment, this is a service which you can purchase from Biosolvay team. It's not yet a software product, clearly under development, but not yet. At the moment, you have to do this in a collaboration with us. And we will come back with a list of molecules that we recommend for your purchasing for you. And as a last thing, I want to talk a little bit about what's our future directions, what are we planning to do. Of course, those spaces always should get bigger, and we are working on making searching times shorter and shorter every day, and equally capturing a big variety of reactions. I have focused here a little bit on the real space, which is selected for being purchasable, so only very reliable reaction and building blocks that are on the shelf. But of course, you can go and take a bit more exotic reaction and bring in bigger variety to have maybe compounds that have a very interesting 3D scaffold or a higher sp3 fraction, whatever you're interested. Plus, we are working on a few methods to predict better which building blocks are maybe reacting. Can we learn something from what we know already? And can we work with more building blocks? What's the maximum what we can feed into those spaces at the moment? So size is clearly a topic, but equally things like targeted spaces. And I want to bring up DNA encoded libraries here. There's many people having a DNA encoded library, but basically no control over what's really in the DNA encoded library, because who can capture the billions of molecules that are in there? No one does it really. So how do you compare those different DNA encoded libraries that you do have? That's possible with our technology as well. It came probably through due to the second poll. We're working on different search capabilities as well. The chemical space docking I've elaborated today, but yeah, many of you miss substructure search methods when they're interested in a particular substructure. They might want to be able to search for this. Equally, yeah, there is no coordinates on a feature tree, so shape-based methods are out of interest for us as well. And there's lots of cool stuff going on, and we hope to present more of this to you in the next webinar. And with this, I would like to close and thank you for your attention.